So to sum that up, RNA polymerase is the one transcribing DNA into RNA. The promoter is this green region that the RNA polymerase has to bind, and the promoter is DNA. The enhancer is another area of DNA, often quite far away from the gene, and that's what the transcription factors bind to. And the transcription factors are these purple and green molecules. And it's important to note, you're going to want to add this to your annotation, that transcription factors can activate or repress transcription. In this particular diagram, we are seeing activators because they're helping activate the RNA polymerase. But there are other types that will repress RNA polymerase's activity and turn genes off. Another type of control is our alternative splicing, and we're familiar with this as well. Remember that in a DNA gene, not all of the sequences will end up in the final RNA. So the DNA gets transcribed into RNA, but only the exons, 1, 2, 3, 4, and 5, end up in the final RNA molecule. The introns, these non-coding regions, get cut out and discarded. And the cool thing about splicing is that you can splice the exons together in different ways. For this mRNA molecule, we've spliced together 1, 2, 3, and 5 to get a particular mRNA molecule. In this one, we've spliced together 1, 2, 4, and 5. So the cool thing about this is with one DNA sequence, we can get multiple RNAs, which means multiple proteins. And this helps explain some of the variation in phenotypes that we see within humans or with humans compared to chimpanzees. Because even if two different people have the same DNA sequence, you might get different traits depending on how the RNA gets spliced together.